This series of tutorials will focus less on the biochemical concepts of genetics and more on the methods used to interpret DNA, and how these methods have come to prevalence in modern medical research. As you may already know, DNA is the substance that provides a readable message within the nucleus of each cell. DNA provides a set of instructions which determine the behavior of each individual cell. Before covering some of the more complicated material, I'm going to revise some of the material that you may already have covered, just to make sure that we're on the same page. If the genetic message was to be compared to a book, a nucleotide, which is a single unit of DNA, would be equivalent to a letter. It is clear from this diagram that nucleotides are categorized based on their structure. The similarity between A and G and T and C is evident. Notice the two-ringed structure of the purines and the one-ringed structure of the pyrimidines. This is an important concept when considering the likelihood of one nucleotide mutating to another, but we'll discuss that later. A, which stands for adenine, specifically binds T, which stands for thymine, with 2 hydrogen bonds. And G, which stands for guanine, binds C, which stands for cytosine, with 3 hydrogen bonds. The difference in the number of hydrogen bonds between A and T and G and C makes for a small but relevant difference in the measure of how tightly bound the pairs are. This is another important concept that is taken into account when 2 strands of DNA are separated for various experimental procedures. This diagram depicts DNA's true basic form. Its structure is given the name double helix and resembles a rope ladder that has been twisted repeatedly. In specific regions of a genetic message, triplets, which are 3 nucleotides together, are equivalent to a word when compared to a book. This is because triplets, also known as codons, give rise to specific amino acids in a peptide chain. Such regions of DNA are generally referred to as coding regions. The word code is an important term to remember in genetics. A DNA coding region, commonly known as a gene, is comparable to a paragraph in a book. The coding region is transcribed into mRNA, which is translated into a protein sequence by a ribosome. The mRNA code is made up of introns, which do not code for proteins, and exons, which do code for proteins. Chromosomes are equivalent to chapters in a book. Once you have read all of the chapters, you have finished the book. The entire genetic message within a cell is referred to as a genome. As we progress through this tutorial, the material that we are covering will begin to gain more complexity. It is important to have a good understanding of each concept so that you may have a well-rounded view of genetics. In a multicellular organism such as a human, there are many different cell types, such as skin cells, liver cells, neuronal cells, and many more. Given that all of the cells in a multicellular organism's body are derived from a single paternal sperm cell and a single maternal egg cell, the genome will be unanimous to each individual cell in the organism's body. This may lead you to the question, if the genetic instructions in each individual cell are the same, how do different cell types arise from the same set of instructions? On a general level, the answer lies in the selective suppression of particular genes. This simple diagram depicts the difference between heterochromatin and euchromatin. The word chromatin refers collectively to the DNA and proteins that occupy a nucleus of a cell that is not divided. Heterochromatin refers to a region of DNA that is wrapped tightly by proteins called histones. The tight wrapping of DNA around histones suppresses the genes within, and hence heterochromatic DNA displays a low level of expression. Euchromatin is loose and unbound by histones. Such regions of DNA display high levels of expression. Epigenetics is the study of changes in gene activity caused by factors that are external of the DNA sequence. Epigenetic regulation is what controls the spacing between heterochromatic and euchromatic regions of the genome, and hence which genes are expressed in each particular cell type. With all this in mind, in a muscle cell, genes that code for proteins to allow muscle contraction will be highly expressed, while in neuronal cells, muscle protein expressing genes will lie within the heterochromatic regions of the DNA, as there is no need for these proteins in such cells, and hence are suppressed.